Hello friends, welcome back to my YouTube channel Aston Padasale. In this video, we are going to discuss 100 expected topics for your GATE Biotechnology 2024. First question is, which bacterium is employed in AIMS test? The answer is Salmonella typhimurium is a bacterium which is employed for AIMS test, and specifically histidine auxotrophs of Salmonella are employed for this AIMS test. So in the comment section, please do tell me what is the purpose of the AIMS test. Second question, naturally competent bacteria include Bacillus, Streptococcus, Thermus, Neisseria, Vibrio, Helicobacter, Actinobacter and Haemophilus influenzae. So these are the list of bacteria which comes under the category of naturally competent. So you need to tell me whether E. coli is naturally competent or not. Third question. GenBank is a nucleotide sequence database that is managed by NIH, National Institute of Health. You need to tell me what are the other nucleotide sequence databases. Okay. Fourth question. Template independent DNA polymerases include, there are two template independent polymerase that include terminal deoxynucleotide transferase TDT, and other enzyme include PAP, poly A polymerases which is involved in the process of polyadenylation. Fifth question, lymphocyte interact with foreign antigen in the peripheral lymphoid organ, and that include your lymph node and spleen. Sixth question, which antibiotic leads to premature termination of translation? It is your puromycin. Just try to know more information about this puromycin. Seventh question, which antibiotic mimics amino acyl tRNA? Again, it is your puromycin. So puromycin has a capacity to mimic this amino acyl tRNA, and you need to tell me which amino acyl tRNA is mimicked by this puromycin. Eighth question, maximum insert size for your M13 phage vector is, in otherwise this question will be asked like DNA packaging capacity for this M13 vector, it is 8 to 9 kb. So you need to tell me the DNA packaging capacity of phage meat, BAC, yak and PAC. Okay, because question is going to be asked and you need to know about these things. Okay, ninth question, which organism is employed for commercial production of dextrin? So definitely two questions will be asked like this. Industrial production of citric acid. Which organism is employed for the industrial production of citric acid? Either they will be asking in this form or else match the following type of questions will be asked. When it comes to dextrin, it is produced industrially by Leuconostoid, Lactobacillus and Streptococcus. These three bacterial species employed for commercial production of this dextrin. 10th question. Scrapie is a fatal neurodegenerative disease affecting the nervous system of sheep and goat, and it is caused by perion. Just try to know about other perion diseases infecting human and animal, and just tell me whether there is any genetic material present in this perions, or what does this perion means. Okay, so definitely from this perion one questions will be asked. 11th question, glyoxylate cycle occurs in. So it takes place in peroxisome, and just tell me whether this particular cycle is present in humans and animal, because this cycle is only present in plant. So you need to tell me whether it is also present in humans and animals or not. 12th question, glycerol or in otherwise glycerin is produced commercially by, it is commercially produced both by yeast and green algae. Yeast includes Saccharomyces cerevisiae and Candida species, and your green algae include this particular species. 13th question. Host cell receptor for HIV is, it is your CD4 protein. Okay, so HIV, they will not infect all the cells of the body. They specifically infect your immune cell like CD4 T helper cells and your macrophages. 14th question. O-linked glycosylation. In O-linked glycosylation, the carbohydrate sugar is attached to the oxygen atom of serine or threonine residues in protein. What about N-linked glycosylation? In the comment section, you need to tell me. In N-linked glycosylation, the carbohydrate sugar is attached to which amino acid residues? Okay. 15th question. The restriction endonuclease that produces the same nucleotide sequence but have a different recognition sequence is called as, it is called as isocardamers. Okay, and just try to know more about the example of isocardamer and also about this restriction endonuclease. 16th question, Yamanaka factor. So these factors are generally employed for producing or creating induced pluripotent stem cells, and these Yamanaka factor include OCT3 or 4, SOC2, KLF4 and CMYC. These are the protein transcription factor that are considered as Yamanaka factor. Okay, and just try to read about this Yamanaka factor also, friends. Okay, 17th question. In zinc finger protein, the amino acid residues that coordinate zinc are, it is your cysteine and histidine. Okay, 18th question. Chemical that have been used to induce protoplast fusion. Okay, it includes sodium nitrate. So please know the chemical formula. Another thing include polyethylene glycol, calcium ion and polyvinyl alcohol. So these are the chemical that is employed as a fusogen. Other thing include sometimes they will be employing inactivated Sendai virus for this protoplast fusion also. 19th question. Humanized antibody is one in which CDR will be taken from the mouse and rest of the antibody will be from human. The other name for CDR is that hypervariable region or variable region. So the full form of CDR is complementary determining region. So the variable region will be taken from mouse and constant region will be from human. 20. Question number 20 says identification of blood group involve agglutination. So you need to know about the difference between agglutination and precipitation. In agglutination, insoluble antigen or particular antigen will be employed, and in your precipitation antigen, soluble antigen will be employed. 21st question, an isotype is a class of antibody that is determined by its, so isotype means you need to remember the word heavy chain constant region. So you need to tell me about this idiotype. So the example of isotype include IgA antibody, IgG antibody, IgM, IgD and IgE. Okay, so they differ by their heavy chain constant region. 22nd. In mismatch correction repair system, the parental DNA strand is distinguished from the daughter strand by the process called methylation. 20th. The target enzyme for this herbicide bromoxynil is nitrylase. So you need to tell me the target enzyme for other herbicides also. You need to know about the target enzyme for other herbicides also. Okay. So don't forget to read about that particular thing. Next, with 24th question, which antibiotic inhibit DNA gyrase or which antibiotic inhibit bacterial topoisomerase? It is nalidixic acid, which comes under the category of quinolones. Okay, so it is a type of fluoroquinolones and it will be inhibiting your DNA gyrase, or in otherwise it will be inhibiting topoisomerase 2 and topoisomerase 4. 25th question, the RNA polymerase synthesized during the process of replication in bacteria. So you need to know whether they are asking about bacteria or eukaryote. So in bacteria it is removed by RNA pol1. What about in eukaryote? In eukaryote RNA polymerase is removed by which enzyme? You need to tell me. 26th question, T cell that recognize lipid antigen associated with your CD1 molecules. Okay. So just remember lipid antigen means you need to remember CD1 molecule. 27th question, endogenous antigen are presented onto the surface along with, so endogenous antigen means you need to remember class 2 MHC and exogenous antigen means it is class 1 MHC. 28th question, protein A which has a strong affinity for the Fc region of immunoglobulin is isolated from. So protein A is also employed for the process of affinity chromatography and it is isolated from Staphylococcus aureus. Okay, next 29th question, amino acid residues which is most likely to be found in the interior of water soluble globular protein is mostly inside there will be a presence of non-polar hydrophobic amino acid and it include phenylalanine, isoleucine, tryptophan, leucine, methionine, valine. So these amino acids are generally present inside and you need to tell me what kind of amino acid will be present or what amino acid are present on the surface of the water soluble globular protein. Question number 30, major antibodies in external secretion like your mucus, saliva or is a major antibody in external secretion like bronchus mucusus, IgA, and IgA has unique characteristics. So just read about IgA also. I already discussed in the immunology portion I had uploaded last week. Maybe if you want the link, I will be telling you. I had discussed the previous year questions for this immunology both for GATE DT and GATE Excel. Okay. So in that discuss, I had a lot of questions has been discussed from immunology. So just give a read because from immunology, more number of questions are asking in these days for this GATE Biotech. 31st question, bacteria that have flagella that is uniformly distributed over the surface is called as Peritrichus, and just you need to tell me what are the example of peritrichus bacteria. Okay, 32nd question. Okay, so for this question you need to give me the answer. The shortest phase in the mammalian cell cycle is whether it is M phase or genophase. So you are going to tell me in the comment section. Okay, so I'm leaving this question. 33rd question, maltose. So you need to know about the glycosidic linkage in various disaccharide molecules like maltose, lactose, cellulose, sucrose. Okay, so if possible, just tell me the answer in comment section also, so that many students will be getting benefited. Maltose consists of two molecules of glucose that are linked by. So in maltose, alpha 1,4 glycosidic linkage is present. What about lactose, sucrose, cellulose, cellobiose? You need to tell me. 34th question, hypertrichosis of ear, webbed toes and porcupine mern are example of, it is an example of Y-linked inheritance. Okay. Next thing, 35th question, Gus and reporter gene is isolated from. So Gus reporter gene is isolated from E. coli. Okay. So 36th question, which hormone prevents? So you need to know whether they are asking which hormone prevents or which hormone promote. So you need to read about the questions correctly. So don't make silly mistakes like this. Which hormone prevents precocious germination or vivipary? So vivipary is inhibited by your abscisic acid. Okay. 37th question, which amino acid is holding 3 ionizable group? The correct answer is lysine. And you need to tell me which amino acid has no ionizable group at all. 38th question, which RNA is involved in protecting the germline from transposable element? It is your PV interacting RNA, in short form it is called as piRNA. 39th question, which amino acid is catalyzed by activated macrophages to produce reactive nitrogen species like nitrous oxide? So it is arginine. Arginine is a precursor for producing this RNA. So don't confuse arginine with aspartic acid or aspartate. Okay, arginine is different, aspartate is different. Okay, so you are going to tell me in N-linked glycosylation which amino acid is employed. Okay, so don't confuse aspartate with arginine. 40th question, Kala-Azar is also called as black fever. It is a disease that is caused by a protozoan parasite called Leishmania donovani. Okay. 41st question, which enzyme is coded by ABO blood group locus in human genome? Okay, so this ABO blood group locus is encoding an enzyme called as galactosyl transferase. Okay, so that's why if you take A antigen, it will be coding galactosyl transferase A. That will be adding N-acetyl galactosamine to the H antigen. Whereas if you take B allele, that is B blood group or B antigen, it will be encoding galactosyl transferase B. That will be adding galactose to the H antigen. Okay, so A, B are the different allelic form of this particular locus. Okay, next, which one of the following media component used to maintain the pH in mammalian cell culture? It is sodium bicarbonate, and don't forget to note down the chemical formula of sodium bicarbonate. The sodium bicarbonate is employed to maintain the pH in mammalian cell culture. 43rd, question number 43rd is that synthetic cytokinin include your BAP, 6-benzyl aminopurine and also kinetin. The other name of kinetin is 6-perfurate aminopurine. So you are going to tell me whether zeatinine is the synthetic cytokine or whether it is a natural cytokinin and from which source zeatinine is isolated. You are going to tell me. 44. Question number 44 is coronavirus genome. So coronavirus, either from vaccine you can expect question or with the genome you can expect question. Okay, coronavirus consists of positive sense, single stranded RNA genome, and sometimes you can expect a question with regarding to genome size, and the genome size of coronavirus will be ranging from 26 kb to 31 kb. Okay, 45. Question number 45. Which RNA polymerase is involved in the synthesis of small interfering RNA, that is siRNA in plant? It is your RNA pol 4. So you need to tell me which RNA is involved in transcribing your transfer RNA. Okay. 46. CRISPR-Cas. So you need to tell me what is the full form of CRISPR-Cas in the comment section. So each one you need to tell me. So CRISPR-Cas is associated with, so CRISPR-Cas was first observed in the E. coli only. So it is associated with prokaryote adaptive immunity. Okay, which is an immunity that is protecting against viruses. Okay, 47, the process by which extracellular fluid and solutes are taken up into the cell by a small vesicle is called as pinocytosis, and pinocytosis is an active energy requiring process and it is a kind of endocytosis process. Okay, 48th question. Single membrane enclosed organelle include lysosome, sphaerosome. Sphaerosome is a part of your endoplasmic reticulum. It is just like a lysosome which is present in plant. Okay, so sphaerosome is a plant-based lysosome and your glyoxysome. So you need to tell me which organ comes in a double membrane and which organ doesn't have a membrane at all. 49, question number 49, which database is used for submitting your microarray data and RNA sequencing data? It is expression omnibus. Okay, so in this database only you can find more information about gene expression profile. So they will be uploading microarray data to this database and RNA sequencing data will also be uploaded in this GEO. So you are going to tell me about OMIM. What does that particular database means? So I had asked many questions in this video. So each one should give me the answer. Only then you can score more marks in this GATE Biotech. Okay, cheer up. Last question. Which database is used for alignment of protein sequence? So you need to remember whether they are asking about protein sequence or protein structure. Here they are asking about which database is used for alignment of protein structure. It is DALI. DALI is a specific program, bioinformatics program that is employed for aligning your protein structure. So up to this I'm stopping. So next 50 question will be uploaded in part 2. Just wait for the part 2 video. Thank you friends. Thanks for watching this video.